So, can we use the sort of analysis we've taken to identify sequence relationships between organisms, and you turn that into an estimate of evolutionary relatedness and time of divergence during evolution? But before we can do that, we've got to make some adjustments to our assumptions in doing the analysis in order to calculate genetic distance with some degree of reliability. And I'm going to tell you about three different caveats you need to take into account. First of all, it's known that not all mutations are equal. So, for example, the mutation which changes a C into a T from one pyrimidine to another pyrimidine, which is called a transition, happens at a higher frequency. It's an easier event than a transversion, where a pyrimidine changes to a purine, or vice versa. Now, we know the relative frequencies at which those can happen spontaneously, and so we can ask our computer algorithm to compensate appropriately. Then, because there's only four nucleotides that can exist at each position, if the mutation rate goes up to a certain level where a position can be hit more than once by a mutation, it's possible for us to start underestimating the number of mutations that have taken place between two different organisms, because a sequence can have previously been, for example, A, mutated to G, mutated again to C, and mutate again back to A, so there have been actually three different mutation events at that position, but it looks like there have been none. Now we can make some reasonable estimates of the degree to which we're underestimating genetic distance based on multiple hits, based on simple statistical distributions. And finally, and most importantly, we have to make sure that we align the sequences that we're going to use in this analysis correctly before we start doing any of those procedures like maximum parsimony or likelihood. So I'm illustrating that with a little group of sequences here. On the left, we've taken that group of four sequences and simply started them all off the same 5' position and written them against each other and highlighted in magenta where the nucleotides of the sequences differ from the reference sequence, the first sequence. And you can see there's quite a large number of magenta nucleotides in that whole population. But if we simply allow those four sequences to slip relative to each other, we find that there's a position at which they can slip to, which gives us much lower levels of nucleotide substitution. In fact, in this case, if we just allow them to align to their 3' positions, we've reduced the number of mismatches between the sequences very substantially, down to just 4 in this case. But that's not all we can do. In addition to allowing sequences to slip globally past each other, we can also recognize that sometimes insertions or deletions occur in sequences, really quite common. And so if we allow insertions or deletions, or indels they're called, to introduce gaps into these sequences, we can get them much more closely aligned. But of course, we have to be careful not to introduce too many gaps, because otherwise everything aligns perfectly. So we need to allow a certain penalty for the insertion of gaps in our sequence alignment algorithms. All of these considerations are put together in a correction matrix that the computer algorithm uses when it's doing its sequence analysis to try and get us the best chance of having a reliable estimate of the genetic distance between sequences. So here's a little example I did earlier on today with one deliberate mistake in it. So here are a bunch of amino acid sequences, in this case, of the alpha globin gene. So alpha globin is one of the components of hemoglobin. Just showing you a snapshot of part of the amino acid sequence there, and I have aligned them to each other in this program. In this case, the insertions and deletions, the gaps introduced by the alignment, are shown up as hyphens. There's one just there at position 49 in the human sequence. And the dots are not gaps, they are representing identical sequences with the reference, the human sequence, on the top line. And that is just a way of displaying it so you can see more easily where the changes in amino acid are in those sequences. So I've then taken that aligned set of sequences and put it through a program that uses the maximum likelihood method. It's a different method from the one that we showed in the last module, the maximum likelihood method of estimating genetic distances and relationships. And it comes up with this sort of family tree. In this tree, the length, the horizontal length of the branches is proportional to genetic distance, and that bar is equal to 100 substitutions. Now, in many ways, this is very often the case, that when you do this sort of thing, you get relationships between the organisms carrying the genes that make quite a lot of sense in biological terms. So, for example, the great apes, humans, chimps and orangutans are all very closely clustered together, and the next closest relatives is a primate, and then the next closest relatives are the other mammals, and the amphibians are grouped together separately, and the bird is separate, and the fish is separate. But you'll notice the goat is out there more or less on its own, connected by a very long branch from the carp. And the question is, what is it doing there? Is the goat a fish? Is there something funny about alpha globin in the goat? Or have I made a mistake? And I thought I'd leave this in because it's a good example of how careful you have to be. Because you have to pick the right sequences to align and compare against each other. Because quite often genes duplicate in evolution, and you have a bunch of different genes that have similar evolutionary origins, and they each drift apart during evolution. And this is the case in the globin cluster. The alpha and beta chains were originally one gene, but they duplicated early on in evolution and then had separate evolutionary trajectories. And what I've done is I've attempted to align the goat beta globin gene with the other alpha globin genes. And of course I put a nonsense result. We can use analysis such as this to calculate a molecular clock, or we can ask the question, can this sort of information give us a useful molecular clock? And this is one of the earliest studies doing this sort of thing, where a large number of mammalian species were used. 17 different genes within each of those mammals was analyzed, and the genetic distances between those genes was averaged across those different genes, and that's on the y-axis. And on the x-axis was, at that time, what was best known as the evolutionary separation of those different species based on the fossil record, where the last common ancestor of the pairs of species had been estimated to have lived. And what you can see is you plot the last common ancestor from the fossil record against the percentage nucleotide substitution, you get a pretty good straight line. And it's using that sort of information that enables people to generate molecular clocks, so that if we were to come across another mammalian species, we would be able to use this information to say what were its closest relatives and how long ago did it diverge from those relatives, with a fair degree of precision. But the caveats for this clock, even when we've taken all the best precautions that I've described to you already, are that different genes show different rates of evolution. So we have to make sure that we use well-calibrated gene sets, and preferably multiple gene sets, in order to do these clocks. We also know that rates change substantially over time and place. So we can't use this relationship between substitutions and time in order to estimate the time of divergence of something that's very, very much more anciently diverged from the group that's shown here. And you always need some independent way of calibrating the sequence substitution rate independently of the sequences themselves, if you want to be able to tie it